Hello students, in the last video in the biotechnology in application chapter we have started the use of biotechnology in the field of medicine. So in that we have seen how to obtain the genetically engineered insulin. We have completed that part. Then after that we will see the next part in the same field, that is in the field of medicine, that is to obtain the therapeutic agents. So what are the therapeutic agents? The people, those who are suffering from different type of diseases, if it is related to hormone deficiency type of disease or immune related disease also, all these diseases can be treated. It is possible by just the injection of some type of hormone, some type of proteins. The diseases can be treated and it is possible. Suppose some people they are suffering from dwarfism, then any medicine is not going to help that individual to grow, that child to grow. So in that cases some type of hormones injected, and this way the hormone, if it is injected into the body, the child will have the normal proper growth. So these are actually the therapeutic agents, the chemicals that are suppose hormonal, this type of proteins that can be used to treat different kinds of diseases. So let us see all the type of therapeutic agents. We already know these therapeutic agents, those proteins. These proteins will be formed from a specific gene. So that gene will undergo transcription to produce a mRNA, and after that it will produce the protein by the process of translation. So what happened, that specific protein or the specific enzyme producing gene, if we can insert into the E. coli with the help of a vector, we can make a host, competent transformant host. Then after that this transformant host will produce our required product, that can be a protein or that can be a hormone. So let us see what are those things, how actually we can able to extract the product and what are those products actually. So first of all, here it is given, that is HGH. HGH stands for human growth hormone. So human growth hormone has obtained for introducing HGH gene in E. coli to treat dwarfism. So what is it? See, suppose this human growth hormone is actually a protein. Due to the lack of human growth hormone, the individual will suffer from dwarfism. Now, what the biotechnology made possible, that this human growth hormone producing gene, okay, so this is the gene, that gene is introduced into the E. coli. So this E. coli, by the translation process, that will produce human growth hormone hormone. So this hormone will be injected to that individual, and that individual will attain the normal growth. So this way it can be treated. The people they are suffering from dwarfism, it can be treated by this biotechnological use, that is by human growth hormone, that is not produced by the normal process, or that individual by himself he cannot produce human growth hormone. So that's why it is synthesized artificially with the help of biotechnology to treat dwarfism. The next one, that is TPA. TPA stands for tissue plasminogen activator. See, this is important. Tissue plasminogen activator. Okay, so this tissue plasminogen activator is used as a clot buster. Suppose some people they're having high pressure or stroke, in that condition to treat that individual, because that individual, if the blood starts to clot inside the blood vessel, maybe the blood vessel will rupture and that individual can have stroke. So in that case some injections are given that will have TPA, tissue plasminogen activator, that is also synthesized artificially with the help of biotechnology. Next one, the third example is antihemophilic factor 8. This is actually one factor due to what the individual will have the proper blood clotting mechanism. Now, due to the lack of this antihemophilic factor, that individual will not able to stop blood flow. So once it has been cut, that individual will never stop blood flow, and this way the people, they die. So this is one genetic disorder, that is hemophilia. You will get it whenever you will study molecular basis of inheritance. So see, in this antihemophilic factor that is used artificially, it is synthesized and it is used to treat a disease that is called as hemophilia. So hemophilia is a disease where the individual cannot stop blood flow. If it has been cut, the blood vessels are cut, then continuously blood will release from the body. So excessive release of the blood from the body can lead to death. So this is the disease hemophilia. This is a genetic disorder. The people, they are born with the disease hemophilia that can be treated properly with the help of this artificial antihemophilic factor 8. Next one, that is interferon. Remember, interferon, already I have told you that in case of our body to treat different kinds of viral diseases produce interferons. In the physiological barrier, in the chapter Human Health and Disease, we have studied already all those things, that this interferon is used to treat the number of viral diseases. Like for HIV AIDS also, this interferons can be taken to at least increase the lifespan of that individual. So this is interferon, which is used to treat a number of viral diseases including HIV AIDS. Next one, the next example is very much important, and this one also we have discussed in the Human Health and Disease chapter, that is second generation vaccine. Normally, whatever the first generation vaccines actually are, Edward Jenner, he developed this whole idea of vaccination. He actually took the foreign particle, that is of even the cowpox, that antigens, cowpox causing antigen that is introduced into the people, so that the people develop antibody against it. So this is the first generation vaccine. So first generation vaccine can have lots of advantages, like he may develop some another type of proteins also along with the first generation vaccines. So that's why second generation vaccines have been developed by biotechnology, by which only one type of antigen will be present. No another antigens will be there, and that if it is introduced, the body will develop only that specific antibody which we need to treat only that disease. Okay, so that's why the second generation vaccine have been developed. Next one, another interesting type of vaccinations that have been also developed to actually give the children, the infants, they are the edible vaccines. Edible vaccines means some type of vaccine property is introduced into some fruits like banana. In banana, that banana is introduced such a way that it has the antigens in it, and if the child, they take the edible vaccine or that specific, suppose GMO banana, that the child will get the immunity. So this is the edible vaccine that is also produced by the same process, same technology, that is with the help of biotechnology. Next one, the next example is interleukin. So interleukin is used to stimulate the immune system. If that individual have weak immune system, or if that individual need to activate the immune system, then some type of that is interleukin, which is produced by the biotechnology, it is used to activate or to stimulate the immune system. Next one, that suppose that individual is suffering from any type of hormonal deficiency, or that individual is suffering from, suppose, gigantism, then that individual can have growth inhibiting hormone. Next one, thymosin, which is released from the thymus. If that individual do not produce this thymosin, then what can happen? The cell-mediated immune system will be deactivated. He will not able to develop cytotoxic T cell. So in that case, thymosin can be injected from outside to mature the T lymphocytes. Next one, gonadotropin. If that individual don't have also this gonadotropin hormones, then also the individual will not able to reproductively mature. He will not able to produce the gametes. For all this type of problems, the hormonal deficiency diseases, that can be treated easily by introducing the hormones. So those hormones, they are artificially synthesized in the biotechnology laboratory. Next one, artificial blood. So this time has arisen that artificial blood that can also be synthesized artificially. So what is it? This artificial blood that can be prepared by introducing hemoglobin producing gene in transgenic pig. So in transgenic pig, a gene that is hemoglobin producing gene is introduced. So that pig will produce that blood which will have the similar characteristics, similarities of human blood. So that can be used for the treatment of some suppose that individual is suffering from anemia, or suppose blood loss, or suppose that individual have met accident. For the rare blood groups, this is a solution. So artificial blood can also be a solution for the different kinds of blood or that suffers from anemia, or sudden suppose any accidents where the severe blood loss had occurred. So for the treatment of that also artificial blood can be used. So just see, these are just some examples which is given in your NCERT or which may come in your NEET examination. Apart from that, a number of varieties of therapeutic agents are here which can be useful for the treatment of almost all kinds of diseases. Now also it is growing, and after some time it will be very much common, and no one will suffer from all kinds of hormonal deficiency diseases. So these are all about the therapeutic agents you need to know for your examination. So that's all about 8 of the therapeutic agents you need to know. From that, this TPA is important, human growth hormone is important, interferon, interleukin, artificial blood, these are important. You just see once. That's all about it. I hope you understand. Thank you.